Welcome to the deep dive. Today we're grabbing a stack of sources that honestly look less like current medical research and more like a blueprint for medicine maybe, 20 years from now?
It really does.
We're diving into three massive frontiers. First, how AI is designing completely novel therapeutics. Then, how huge real-world data is changing how we use blockbuster drugs. And finally, the absolute cutting-edge research into actually reversing biological age.
Yeah, this is a deep dive into health span. We're not just talking about managing sickness anymore. We're talking about reprogramming the human system itself.
So what's our mission here?
Our mission is to pull out the most surprising, the most concrete, and the highest impact takeaways from all this. We'll go from tackling chronic organ failure, fibrosis, with incredible specificity,
Right.
to flipping the master switch on chronic inflammation, and then, ultimately resetting our own internal clocks.
Okay, let's unpack this. We have to start with what AI can do that, frankly, human researchers couldn't. It's about identifying and targeting the chronic killer that's responsible for it. I mean, the number is huge, up to 45% of deaths in the industrialized world.
And that killer is fibrosis.
Fibrosis.
It's essentially pathological scarring, excessive scarring. If you have a chronic organ disease, doesn't matter if it's your kidney, your liver, your lungs, something like idiopathic pulmonary fibrosis or IPF. Scarring is the final, deadly stage.
And IPS is particularly nasty.
It is. It's this gradually advancing and almost always lethal disease. Current treatments, they slow it down, but we desperately need new targets to actually reverse it.
So enter this AI platform, Pandaomics.
Yeah.
It scans just mountains of biological data, and it flagged one specific molecule, the #1 target for fibrosis.
Yeah.
Transmembrane-associated kinase, or TNIK.
Mm-hmm.
Why was that so novel?
Because TNIK, which is a serine threonine kinase, was largely uninterrogated, is the term they use.
Uninterrogated, so just ignored.
Pretty much. Ignored by traditional pharma pipelines for IPF. Human researchers, we tend to focus on familiar drug classes, like the tyrosine kinase inhibitors that are already out there. The AI saw the whole system.
And it saw a connection.
It saw that TNIK connects directly to the core pathways that are driving that scarring process.
Okay, hang on. You're getting technical right away, so let's give you, the listener, a quick guide. You mentioned WNT, TGF-beta, and hippo signaling. In simple terms, what are those? Why do they matter for scarring?
Okay, yeah. So think of them this way. They are the chief architects of how your tissues develop and repair themselves.
The architects.
Right. TGF-beta, for instance, is the master regulator. It's the one that tells cells, okay, start building scar tissue. WNT signaling that governs cell fate, how they multiply. And the hippo pathway regulates organ size.
So the fact that TNIK influences all three.
That's the key. It sits upstream, influencing all three of these massive systems. It means it's an incredibly powerful lever to pull to stop that runaway scarring process. The AI found a critical choke point.
That makes so much sense. It's not some minor pathway. It's the intersection of all the scaffolding systems. Okay, so once they had the target, the generative AI designed a small molecule, right? INS-01805.
It did. And the chemistry on this thing is aggressive in a good way. It has a really potent binding affinity, a KD value of 4.32 NIM, which is fantastic.
And the mechanism is unique.
Very. The AI predicted it would occupy this back pocket of TNIK and do so very deeply. It essentially just jams the function of that whole intersection point.
But the real test is always translation. Did it work in a living system?
It absolutely did. They used a standard mouse model of lung fibrosis induced by a chemical, bleomycin, which mimics severe human lung scarring. The INS-01805 treatment. It led to a significant dose-dependent reduction in the fibrotic area, and this is the key number. At high doses, they saw over a 75% reduction in scarring.
Wow, 75%. How does that compare to what's already out there?
That level of efficacy is comparable to the established drug, nintedanib.
So it's not just promising, it's on par with the current standard of care, at least in this model.
Exactly. And beyond that, the initial human data is good too. Phase 0 and Phase I trials in healthy people showed a favorable safety profile. It's well tolerated, it's absorbed rapidly.
So it hits its peak concentration in the body in like an hour and a half.
Right. This whole process, from an AI spotting an overlooked target to designing a unique molecule to then showing equivalent efficacy in models, it's just a huge statement about the speed and novelty of AI-driven medicine.
It's incredible. Okay, so if AI is designing the future, Let's pivot to reevaluating the drugs that are already changing the present. I'm talking about GLP-1 receptor agonists.
Semaglutide, tirzepatide.
Exactly. Everyone knows them for weight loss, but the bigger question, the longevity question is, what is their impact and how do they really stack up head-to-head?
That's the critical shift in thinking, seeing them as longevity agents, not just, weight loss drugs. And we finally have the data. Real-world evidence comparing the cardiovascular outcomes.
Semaglutide versus tirzepatide in patients with obesity and type 2 diabetes.
Right. And we already knew both were better than older drugs. I mean, semaglutide versus, say, sitagliptin gave a hazard ratio of 0.82 for major cardiovascular events. That's a strong benefit.
But the head-to-head is what everyone was waiting for.
And the crucial finding was, when you look at that composite outcome, heart attack, stroke, or all-cause mortality, the cardiovascular benefit between acetamide and semaglutide was comparable, a hazard ratio of 1.06.
So, essentially the same, parity. For you listening, what does that suggest about the GIP component in tirzepatide? Is it maybe not adding as much heart benefit as the GLP-1 part?
That is the exact question researchers are asking now. It really suggests that the primary cardiovascular protection is coming from that GLP-1 mechanism, stabilizing metabolism, improving blood pressure, insulin sensitivity.
Regardless of the second GIP component.
It seems so. The major benefit appears to be shared across the class.
And as longevity agents, that benefit goes way beyond just the heart. They're powerful because they don't just treat obesity, they restore homeostasis. They stabilize those nutrient-sensing pathways. They obliterate visceral fat, and they actively dampen inflammaging, that insidious low-grade chronic inflammation that just damages your tissues over decades.
They're essentially applying the brakes to the aging process. By cutting down on metabolic and oxidative stress, they slow the buildup of those dysfunctional senescent cells, especially in the walls of your blood vessels.
Those are the zombie cells that leak inflammatory factors and stiffen your arteries.
The very same.
Okay, so if GLP-1s are handling the metabolic side of inflammaging, researchers are now looking for the dedicated inflammatory switch. And this brings us to the NLRP3 inflammasome.
Right, which is quickly being called the master switch for inflammaging.
The third pillar of preventative medicine, right?
Right.
Alongside statins and GLP-1s.
That's the idea. The NLRP3 inflammasome is basically your body's molecular security alarm. It detects threats like metabolic junk, crystals, pathogens, and it flips the switch to trigger a massive inflammatory response.
But in age-related disease, that alarm gets stuck in the on position.
Exactly, leading to chronic systemic damage.
And in inhibiting that stuck alarm, it produced some pretty astonishing results in trials.
It really did. Ventyx tested their oral NLRP3 inhibitor, VTX3232, in high-risk patients. They saw a nearly 78% reduction in hsCRP.
That's high-sensitivity C-reactive protein, a key marker of inflammation.
A 78% drop. It's a massive reduction.
But the independence observation here is absolutely crucial. The compound did that, but VTX3232 did not cause weight loss on its own.
And that is the powerful evidence for the third pillar idea. It confirms that the NLRP3 inhibitor is targeting a totally separate pathological axis, pure destructive inflammation, that is distinct from the appetite and insulin axis that GLP-1s hit.
So we need separate tools for separate problems.
We do. And because they're separate, they are complementary.
They work together.
Precisely. The data showed that when you added VTX3232 to semaglutide, you got significant additional drops in inflammatory markers like hsCRP and IL-6. You hit the metabolic driver with the GLP-1, and you extinguished the inflammatory fire with the NLRP3 inhibitor.
And the beauty of targeting that NLRP3 alarm is that it's versatile. Yeah. It's all over the body.
Exactly. It's implicated in so many age-related diseases, not just heart health. I mean, in neurodegenerative disorders like Parkinson's, a brain-penetrant inhibitor, NT0796, it significantly reduced inflammatory biomarkers right there in the cerebrospinal fluid in just 28 days.
Wow. So it validates the whole idea of reducing chronic inflammation is central to maintaining function across every single system.
It really does.
Okay, let's go even deeper. Let's transition to something more profound. We're moving beyond controlling symptoms or even inflammation to fundamentally altering biological time. Let's talk about interventions that can actively roll back the clock.
Right. And we can start with some surprising age reversal data from, of all things, metformin.
In monkeys, right?
Yes, in old male cynomolgus monkeys. Scientists measured their biological age using these multi-omics clocks, looking at DNA methylation, gene expression.
But really deep biological markers.
Very deep. And metformin treatment showed a genuine deceleration, even a reversal, of aging across major metabolic organs.
And the results were quantifiable. It reduced the biological age of the liver by about 4 years and the kidney cortex by around 3.4 years.
You could almost say the monkeys got a biological software update for their organs. It wasn't just managing blood sugar, it was repressing the actual pathways tied to aging, inflammation, cell death, fibrosis, while reactivating youthful pathways like DNA repair.
This feeds directly into this modern theory of aging, that it's really a loss of epigenetic information.
It is.
And there's this fascinating mouse study that just drives this point home. Researchers engineered mice so they would get DNA double-strand breaks, literally just cutting the DNA structure. But without causing the mutations, we linked to cancer. They called it the ICE system.
And the discovery was profound. Just by creating those breaks, that structural corruption of the DNA, the mice aged. They aged roughly 50% faster than the control group.
50% faster?
Yeah. And they developed every hallmark of old age. Hair loss, a stooped posture. They moved less, their memory got worse.
So the big takeaway for you here is that aging might be less about accumulated damage like mutations and more about accumulated corruption of the epigenetic code, the instructions on how to use the DNA.
Precisely. The system loses its ability to read the code correctly. But the good news is, if you can cause aging that fast by scrambling the instructions,
You might be able to reverse it.
and they did. They used what are called partial reprogramming factors, specifically OSK, which is a simplified version of the Nobel-winning Yamanaka factors.
And when they gave that to the prematurely aged mice,
It rejuvenated the aging markers in their muscle and kidney tissue. It essentially returned them to a youthful state. The information wasn't gone, it was just recoverable.
that is just molecular level recalibration. Okay, let's zoom out one last time. From the molecular to the entire systemic environment controlled by our blood plasma. This might be the most counterintuitive, profound finding in this entire dive.
It's highly provocative because it flips the entire script. The research shows that if you take old blood serum and mix it with young cells, the old blood serum completely dominates.
The old blood wins.
It wins. It actually induces senescence, that zombie cells stayed in young tissues, in just 2 weeks. The old factors are actively inhibitory. They're dominant.
So the therapy here isn't about adding youth. It's about clearing out the old.
Exactly. The researchers found that a large dilution of old blood plasma, just replacing 50% of it with a simple saline albumin solution, robustly rejuvenated cells and tissues in old mice.
Across all 3 germ layers.
Yeah.
So the whole body.
The whole body. By simply diluting the inhibitory factors, the young regenerative factors that were already there could resurface and get back to work.
And this wasn't just some general wellness boost, right? It had specific neurological benefits that other treatments didn't.
Critically, yes. Plasma dilution improved hippocampal neurogenesis, that's the growth of new brain cells, and it significantly diminished neuroinflammation in old brains. These specific cognitive benefits were not replicated by a known potent senolytic molecule.
ABT263.
Right, which confirms that this systemic dilution is addressing a separate and very powerful axis of aging than just clearing out old cells.
That is a staggering finding. Okay, so to round this out, let's touch on one final mechanism for clearing those cells, a highly specific senolytic.
Right, so we have the discovery of miR-106b5P. It's a senolytic microRNA. Remember those senescent zombie cells we talked about?
Yeah.
This microRNA selectively targets and eliminates them by inducing apoptosis, which is controlled cell death.
And how does it do that?
Well, it works by enhancing p53 acetylation and upregulating PUMA. You can think of p53 as the cell's internal quality control inspector and also its suicide switch if things go wrong. PUMA is the protein that actually carries out that order.
So the microRNA basically just flips the switch on the bad cells.
It enhances the quality control function, telling the senescent cells, okay, it's time to shut down for good. And when they delivered this systemically in aged mice, it reduced senescent markers and inflammation across multiple tissues.
What a sweep of the future of health. So for you, the listener, you now have insights spanning from these hyper-specific AI-generated molecules hitting previously ignored targets for organ failure, all the way to a global understanding of metabolic and inflammatory control with GLP-1s and NLRP3 inhibitors, and then finally these fundamental interventions that can recalibrate the entire physiology of aging. It's a remarkable picture.
It is, and this leads to the most provocative thought for you to carry forward, and it comes from those systemic studies, the findings from blood dilution. The idea that aging is driven by potent inhibitors in our blood that actively suppress youth factors. It suggests aging isn't just accumulated irreversible damage, it's a phenotype. It's a state that's controlled by our systemic environment.
Which raises a really important question.
It does. If aging is primarily driven by elevated inhibitory factors, rather than a simple lack of restorative ones, should anti-aging research focus more heavily on lifelong systemic management and clearance like dilution to remove these inhibitors, instead of just constantly trying to add more and more restorative factors back in? Understanding and managing your systemic environment. That truly might be the key to unlocking your health span.